Okay, so we're going to spend the next bit of time on the urinary system, specifically talking about these units that we call nephrons. And this right here represents one nephron. And the reason these are important is because these are the functional units of the kidneys. Okay, these are going to be responsible for performing the job of processing blood and maintaining blood chemistry. And there's not one of these, there's not 10 of these, there's not 100, there's 1,000,000 of these in each kidney. And specifically, we find these nephrons located up here in the renal cortex. And like I said, these nephrons are going to be responsible for actually processing blood, for maintaining blood chemistry. So let's go through some anatomy of it here. So the first part of the nephron is called the renal corpuscle here. And the renal corpuscle, that's the renal corpuscle, is composed of two different parts. One of those is called the glomerulus. And what the glomerulus is this capillary bed that you see inside of there. And that capillary bed is where fluid is going to be removed from the blood. The second part of the renal corpuscle is called the glomerular capsule. And the capsule is just that. It's a capsule that fits over the glomerulus, the capillary bed, and essentially what it's going to do, it's going to catch that fluid that's being removed from the glomerulus. So the renal corpuscle is where material leaves the blood for the kidney. And in doing so, we're going to produce a new solution that's called filtrate. So fluid's going to leave the blood, it's going to be caught by this glomerular capsule, and that new fluid that we produce is called filtrate. And one thing that I just want to point out before we move on here, just so you can orient yourself where you're at in the kidney, I said it's up here in the renal cortex, but this is a good indicator of that. All of this area above here is the cortex. All of this area below there is the medulla. So you can see that most of the nephron is located up here in the cortex. And by the way, renal corpuscle is not a term that you guys are going to need to know. I may reference it, I may talk about it, but as far as just that being on the exam, that won't be an exam question itself. But you do need to know the individual parts of it, the glomerulus, which is the capillary bed, and the glomerular capsule that fits over it and basically catches that filtrate that's coming out of the glomerular capillary bed. All right, and the second part of the nephron is called the renal tubule. So everything else here is all renal tubule. And there's three different parts to it. This first part is the proximal convoluted tubule. This next part is the loop of Henle, and the last part is the distal convoluted tubule. Now what's going to happen here? So we produce this new solution called filtrate, and now that filtrate is running through this renal tubule here. And what's going to happen is the renal tubule is where most of this filtrate is returned to the blood. So we have this solution traveling through the renal tubule, and most of that is going to be returned back to the blood. Why are we going to return things back to the blood? Because that's how we maintain blood chemistry. We take things out of it, and then we return what we need to the blood to maintain blood chemistry. So as that filtrate travels through the proximal convoluted tubule, we have a lot of substances that are being reabsorbed, being taken back into the blood, and it continues to do that. And as we go through here, there's less and less filtrate because we're returning that stuff to the blood. But basically, by the time it gets to the end of the distal convoluted tubule there, whatever has not been returned to the blood, it drains into these structures right here. Color that orange actually. That we call collecting ducts. And so these collecting ducts are collecting all this filtrate from the nephrons that have not been returned to the blood. And you can see that several nephrons dump their contents into this collecting duct. And once that solution enters the collecting duct, it goes from being called filtrate and it's now urine. So we now have urine that's traveling through those collecting ducts there. But the collecting ducts are not part of the nephron itself. Okay, so let's look at some different steps of all this. So the first thing that we want to look at is glomerular filtration. And basically what we're doing with glomerular filtration is we have blood that's coming into the glomerular capillaries, and we are removing fluid from that blood, and it's being caught by the glomerular capsule there. And then the new name for that fluid that enters the glomerular capsule is called filtrate. So that's glomerular filtration. And this is a more detailed view of actually what's going on there. So here we have the glomeruli, the glomerular capillary beds, and the way you can think about the glomerular capsule is you can think of it like a balloon, and it's kind of like you stick this capillary bed down into that balloon. And so there's a layer of the glomerular capsule that lines the capillary bed itself, and then there's another layer out here that actually forms the outer boundary of the capsule. And so that's what you're looking at here. You can see that a layer of that glomerulus is wrapping around the capillary beds. And that's all this stuff right here. That's all part of the glomerular capsule. And the reason that's important is that collectively the capillary and this portion of the glomerular capsule form what we call a filtration membrane. Okay, we looked at a respiratory membrane, now we're looking at a filtration membrane here. And basically this filtration membrane is going to determine what leaves the capillaries and becomes filtrate. And you guys know from the cardiovascular, you know that capillaries are leaky, you have these pores in them, you have slits in them, you have all this stuff that allows fluid to move out of there. But now you have this extra element, you have this part of the glomerular capsule over the top of it. So you have these cells and they have these little processes and they form what are called filtration slits. And so only certain things are going to be able to move out of the capillaries through the capillary wall through these filtration slits and become part of the filtrate here. Now, what is this filtration membrane selecting for? Is it selecting for charge? Is it selecting for, what determines what actually moves out of the blood through this filtration membrane and becomes part of the filtrate? And the answer is size here. We're selecting for size. And specifically, are small or large molecules going to be able to become part of the filtrate? It's only small molecules that are going to make it through that filtration membrane and become filtrate in the glomerular capsule here. And here's a closer up look of that filtration membrane. Here's the capillary, and they're just simple squamous epithelium, so a really thin cell layer there, but then you have these pores in between it. Then you have a little basement membrane there, and then you have the glomerular capsule, and specifically you have these filtration slits in between there. And so what passes through there? Well, small molecules do, specifically molecules smaller than 3 nanometer in diameter, which is super small. So what does that mean? What does that include? Water. Water's going to pass through there. Glucose is going to pass through there. Amino acids, ions, vitamins, nitrogenous waste. All these things are small enough that they make it through this filtration membrane into the glomerular capsule and they become filtrate. What doesn't make it through there? Well, large molecules, proteins that we find in the blood, red blood cells. None of those, all of those are too large and they can't make it through that membrane, which is good because we don't want to lose these things, okay? We want to keep those in the blood. And you can think of this like a coffee filter. You know, you grind up grounds and then you add water and it dissolves all those grounds, and then here's your there's your membrane, your coffee filter, and what's going to pass through that membrane? Small things are. What's not going to pass through there? Large things like these, like the coffee grounds. And so this fluid right there is analogous to the filtrate that's being formed. Now, if there's compromising of this capillary bed or this membrane, then things that normally don't pass through are going to be able to pass through. And diabetes is something that can cause problems here. You remember with diabetes, what's characteristic of diabetes? Hyperglycemia. And if you remember, high blood sugar levels damage the blood vessels of the body, and specifically they especially damage the capillary beds. So they can damage these capillary beds in the kidney and they can become scarred, which is what you're looking at right there. And the problem with that is that now the filtration membrane is compromised, and so now you can have these large particles that normally don't pass through there. They can become part of the filtrate and they can end up in the urine. This is another picture of that, and here's, you know, your glomerulus. But something I want to point out, because we're going to have to learn some of the blood vessels that are involved here. The glomerulus is fed by a blood vessel called the afferent arteriole, and then that blood goes in there, and it's drained by a vessel called the efferent arteriole. Okay, what does AF refer to? AF means toward. What does F refer to? It means away from. Okay, so this arteriole up here is moving towards something. This arteriole down here is moving away from something. Okay, so what is this relative to? What are we moving toward and away from? We're talking about the glomerulus here, the capillary bed. The afferent arteriole goes towards that capillary bed. The efferent arteriole goes away from it. And then what you can see here is that fluid becomes filtrate down there, and now it's in the glomerular capsule. The next step that we have going on here is something called tubular reabsorption. So now we'll look at this schematic here. This is just a simplified schematic. But what we looked at there was glomerular filtration. We're producing a new fluid called filtrate that goes into the glomerular capsule. Well, next, that fluid goes into the renal tubule. Okay, the proximal convoluted tubule, loop of Henle, and distal convoluted tubule. And this is where tubular reabsorption is going to occur. And what tubular reabsorption refers to is returning things to the blood. So we just took things out of the blood in the filtrate. Now we're going to return them to the blood. And specifically, we're going to return those into another capillary bed called the peritubular capillary bed here. Now, why are we returning things to the blood? We take all this stuff out of the blood, and then most of that we're actually going to return. Why are we doing that? We're doing that to maintain blood chemistry. And essentially, we're going to return almost everything. All of the glucose, all of the amino acids, 99% of the water about, and salts. is going to be returned to the blood. And again, we're doing that to maintain blood chemistry. Anything that does not get returned there leaves the body as urine. Okay. Now this diagram over here is a little better for that. Here we have an afferent arteriole that feeds the capillary bed, and it's drained by an efferent arteriole. And then let's take a look. There's the glomerular capsule, and here is all the renal tubule. And I want to point out here, this is important to note, is that the efferent arteriole becomes this second capillary bed. All of this vasculature right there that's wrapped around the renal tubule, that's the peritubular capillaries. And that's where substances are going to be returned to the blood. So as this filtrate moves through the renal tubule, it's returning most of those substances back into the peritubular capillaries. And basically at that point it's back in the blood. Okay, it's there to do its job. Okay, and this is now a more detailed version of all this. But again, you need to be able to recognize the stuff that's going on here. And I'll point this stuff out. So first of all, this up here is the cortex. This down here is the medulla. And there's blood vessels, there's arteries and veins that run along that border between the cortex and the medulla. And here's where you guys have to start paying attention to this. So there's a blood vessel that comes off of that. I'll use that color. There's a blood vessel that's similar to that. Just go back to this. So there's a blood vessel that comes off these guys down here, and basically these blood vessels here that I'm getting at, these are called the cortical radiate arteries. And what they're doing is, first of all, they're called cortical because they're up here in the cortex. They're up here in the renal cortex. They're called radiate because they're radiating straight up through the renal cortex. And then they're an artery. So you have the cortical radiate arteries, and then you have little branches off of that. And those little branches, they have that guy labeled, are the afferent arterioles. So you have cortical radiate arteries, and then you have these afferent arterioles that come off of that. And those afferent arterioles feed into the glomerular capillary bed. The glomerular capillary bed is then drained by the efferent arterioles. And then those efferent arterioles become these peritubular capillaries that wrap around the renal tubule. And then after all that's done, they basically go back into the venous system. So that's the blood system that you guys need to know in this scenario here. Now let's take a look at the nephron. So here we have the renal corpuscle, the glomerulus and the glomerular capsule. And then this next part is all the proximal convoluted tubule. And then the proximal convoluted tubule becomes the loop of Henle, and then the loop of Henle becomes the distal convoluted tubule. And then that distal convoluted tubule dumps its contents, whatever's not returned to the blood, it dumps it into this collecting duct here. And again, this collecting duct is receiving what is now called urine from several different nephrons. So what's going to happen is we have filtrate that's formed here, and that filtrate travels through the proximal convoluted tubule, and what happens there is that substances are returned to the blood. They're returned to the paratubular capillaries, and now they're back. Then they return to the venous system. Then they're just back in the blood. Okay, and that happens all throughout the renal tubule here. Well, at least the proximal convoluted tubule and the distal convoluted tubule is where this reabsorption is happening at. Now, this is kind of a unique blood pathway system. Normally, what happens is you have an artery that feeds a capillary bed, and it's drained by the venous system. What we have here is we have two sets of capillary beds in a row, and when that happens, you call that a portal system. So the artery comes in, and the afferent arteriole feeds a glomerular capillary bed. Then you have the efferent arteriole, and the efferent arteriole feeds another capillary bed, the paratubular capillaries. So we have two capillary beds in a row before it goes back into that venous system. And this is just a schematic, showing that same thing. Just because, this is all important to know. Here's the border between the medulla and the cortex. And these vessels right up there are your cortical radiate arteries. They're up there in the cortex, and they're radiating upwards. And if you look closely, you can see that there's little branches off of those cortical radiate arteries. Those are the afferent arterioles, is what we're looking at there. And then this is just a schematic here. This just shows here we have a branch of the aorta that goes into the kidney, which is the renal artery. Then it branches. We eventually get to that cortical radiate artery, which feeds the afferent arteriole, which feeds the glomerulus, where filtrate is formed. And then it's drained by the efferent arteriole, goes to the paratubular capillaries, and then back into the venous system. So this right here is where filtrate is formed, and if we're going to reabsorb that, it gets reabsorbed back up there into the paratubular capillaries. Anything that does not get reabsorbed leaves the body as urine. So this is pretty amazing. This is how hard the kidneys are working, but the kidneys are producing about 47 gallons of filtrate a day. Okay, you can imagine almost 50 milk jugs just sitting in front of you. That's how much filtrate the kidneys are producing a day. But obviously we don't urinate out 50 gallons of fluid a day. So of this, less than 1% typically leaves the body as urine. So then if we look at urine itself, urine's about 95% water, but really, this can be higher or lower. It can be 92%. It can be 98%. And why would that change? Why, for instance, if normally it's 95, why would we want that to be only 92% of that is water? Well, that's because we're keeping some more water in the body. Okay, we don't want to get rid of as much water. What if that's up to 99% water that we're getting rid of? Well, then maybe we're overhydrated and we need to get rid of more water. So that water percentage can be higher or lower because we're essentially regulating water balance. And then about 5% are solutes, but again, this can be higher or lower. Maybe we want to keep some more solutes in the body, maybe we want to get rid of more. And by doing so, we're regulating electrolyte balance there. All right, so the first place that this filtrate goes is into the proximal convoluted tubule. And the proximal convoluted tubule is by far the most active reabsorber. This is where the vast majority of reabsorption of substances are occurring. All of the glucose gets reabsorbed at the proximal convoluted tubule. All of the amino acids are reabsorbed at the proximal convoluted tubule. Are you supposed to find glucose in the blood? Or sorry, glucose in the urine? No, you're not. That's supposed to be negative. And that's because all of it is supposed to be reabsorbed at the proximal convoluted tubule here. About 65% of the sodium in water are also reabsorbed at the proximal convoluted tubule. And I just put this up here to kind of illustrate how complicated this is, but this is the proximal convoluted tubule. This is the lumen of the tubule, and this is the capillary bed that it has to return things to. And so the proximal convoluted tubule, the wall of it, is made up of this simple cuboidal epithelium there. And so substances, if they're going to go from the lumen of the renal tubule back into the blood, they have to cross this simple cuboidal epithelium to get back into the blood. And remember, I don't know if you remember from 241, but when you look at histology of the kidney, this is what it looks like. And what this is that this is the lumen of these tubules, of the renal tubules. And then they're outlined by simple cuboidal epithelium. And simple cuboidal is really good at absorbing things, at secreting things. And so that's exactly what's going on here. And I put this up here because it's a complicated process. You know, glucose, amino acids, vitamins, it requires transporters to do that. These sodium co-transporters, it involves some ATP indirectly. You know, it's a lot of activity that's going on here for that reabsorption to occur. But essentially what we're doing is we're taking fluid in the lumen there, we're bringing it across those cells, and we're returning it back into the blood supply there. Then that filtrate goes down into the loop of Henle, and I'm not going to talk about the loop of Henle so much, other to say that there's a lot of water movement there, there's a lot of solute movement there, and it's important in creating this osmotic gradient down here in the medulla that we'll come back to. All right, and then we find ourselves in the distal convoluted tubule, and by the time we reach the distal convoluted tubule, there's only about 10% of the original sodium chloride left and about 25% of the original water left. But we still have something to work with. If we want to return more water to the blood, we can do that. If we want to return more sodium to the blood to regulate electrolyte balance, we can do that. So we still have something to work with here. But at this point, most of the reabsorption, well, not surprisingly, it depends on the body's needs, and it's also regulated by hormones. And specifically what's going to happen is in the distal convoluted tubule is where we are going to reabsorb sodium, any of that remaining sodium. And that's going to be under the control of aldosterone, which is produced by the adrenal glands. So that's under endocrine control. After that, that fluid, which is now called urine, goes into the collecting duct. And in the collecting duct, we have one last opportunity to reabsorb water. Let's say we're dehydrated and we want to reabsorb water. Well, the collecting duct is where that's going to happen, and that's under the control of another hormone called anti-diuretic hormone that's released by the posterior pituitary. And that's how we're going to finish up. We're going to talk about why we might want to absorb more sodium, why we might want to reabsorb more water, and the control over that. Okay. All right. So last time we started talking about these nephrons here. These are the functional units of the kidney. There's 1 million of them lined up there in the renal cortex, And these are the individual blood filtration units. These are going to be responsible for maintaining blood chemistry. And the first thing that we looked at was glomerular filtration. And essentially what's happening with glomerular filtration is we're taking fluid from the blood and we're moving that fluid into the nephron. Specifically, we're moving it into the Bowman's capsule there. And the name for this new fluid that we're producing is called filtrate. Then what happens is the filtrate starts moving through the rest of this renal tubule here. And the first place that it goes is the proximal convoluted tubule. And what happens in the proximal convoluted tubule? This is where reabsorption begins. So we're taking filtrate and we're returning it to the blood. Why are we doing that? Why are we returning things to the blood? We're doing it to maintain blood chemistry. And what specifically happens at the proximal convoluted tubule? Well, first of all, the proximal convoluted tubule is by far the most active reabsorber. So this is where the majority of tubular reabsorption occurs, is in the proximal convoluted tubule. All of the glucose, all of the amino acids, all of the vitamins, most of the water, most of the salts are all reabsorbed at the proximal convoluted tubule here. Then that filtrate travels down through the loop of Henle, which we didn't necessarily talk about, and that's fine. And now we have this filtrate that goes to the distal convoluted tubule. And by the time we get to the distal convoluted tubule, there's about 10% of the originally filtered sodium chloride that's remaining. We also have about 25% of the water that we originally filtered is left at the distal convoluted tubule. So by the time that the filtrate reaches this point, most of this reabsorption is going to be dependent on the body's needs at the time, and it's going to be regulated by hormones. And specifically, what's going to happen at the distal convoluted tubule is we have the final opportunity to reabsorb sodium. And that's going to be dependent on the body's needs at the time, and we'll talk about that. It's also going to be regulated by hormones. And specifically, if we want to reabsorb sodium at the distal convoluted tubule, that's going to be dependent on aldosterone. Aldosterone's a hormone. Does anybody know where it's produced? It's produced in the adrenal glands. So this is our final opportunity to reabsorb sodium. And then what happens is that filtrate enters the collecting ducts, and at this point we call it urine. And once that urine is in the collecting duct, the final thing that we can do at this point is we can reabsorb water. If the body needs water, if we're dehydrated and we need water, this is going to be our final opportunity to reabsorb water into the blood. And whether or not we do this is going to be dependent on the presence or absence of anti-diuretic hormone. An anti-diuretic hormone is produced up in the pituitary gland. So let's start here with in the distal convoluted tubule in the reabsorption of sodium. Okay. So again, this is going to be dependent on aldosterone, and aldosterone is produced by the adrenal gland here. The adrenal gland has a couple different parts. It has an adrenal cortex, an outer layer. Then it has an inner layer, the adrenal medulla. And if you remember from 241, the sympathetic nervous system during the fight or flight response comes down and it innervates, it zaps the adrenal gland. And specifically, it synapses on the adrenal medulla. The adrenal medulla is mostly nervous tissue. And what does the adrenal medulla do in response? It releases norepinephrine or noradrenaline. And that circulates around. It increases heart rate, vasoconstricts blood vessels, bronchodilates, does all that kind of stuff that's associated with the fight or flight response. But then you have this area up here called the adrenal cortex. And this is part of the endocrine system up here. This is going to produce hormones. And specifically, this is where aldosterone is going to be produced, up here in the adrenal cortex of the adrenal gland. Cortisol and testosterone are also produced in the adrenal cortex here. Okay, and this is when that happens. This whole diagram here is called the renin, angiotensin, aldosterone mechanism. And if you look down here, you can see the adrenal cortex releasing aldosterone and acting on the kidney. And why does that happen? Well, we're going to look at all that. So first of all, let's look at the stimulus for this entire mechanism. What stimulates this entire mechanism to take place? A drop in blood pressure. So a drop in blood pressure is going to stimulate the renin, angiotensin, aldosterone mechanism. And we're going to go through all these steps, but the final thing that we want to do is we want to elevate blood pressure. We have a drop in blood pressure, so we want to elevate it. And all this stuff in between is how we're going to do that. And that's the renin, angiotensin, aldosterone mechanism. So first of all, when we have a drop in blood pressure, that's detected, among other places, by the kidney. And in response to that, the kidney releases an enzyme called renin. And renin goes into the blood, and what it does is it converts this molecule called angiotensinogen that's produced by the liver, and it converts it to a molecule called angiotensin 1. Then that angiotensin 1 goes throughout the blood, and when it gets to the pulmonary capillaries up in the lungs, it gets exposed to an enzyme called angiotensin converting enzyme, or ACE. And angiotensin converting enzyme converts angiotensin 1 to angiotensin 2. And angiotensin 2 is a very important molecule in the body. Angiotensin 2 is a potent elevator of blood pressure. And there's 3 different ways that this is going to happen. So remember, we have a drop in blood pressure, and so we want to elevate it. The way that we're going to do that is by producing this angiotensin 2 molecule. And the production of angiotensin 2 involves the kidney, the liver, and the lungs. But now we have this angiotensin 2 molecule circulating around in the blood. And what does it do? One of the things that it does is it acts on the adrenal cortex, and it causes the release of aldosterone. Aldosterone circulates around in the blood, and then it comes back and acts on the kidneys. Where specifically does it act? It acts on the distal convoluted tubule. And what happens when aldosterone acts on the distal convoluted tubule? It takes sodium that's in the filtrate, and it causes it to be reabsorbed, to return to the blood. And you're thinking, okay, that's fine, but how does that help us with low blood pressure? Well, something that's important to know is that wherever sodium moves in the body, water follows along with it. It has to do with the properties of osmosis. But wherever sodium goes, water goes. So if we reabsorb sodium, then we're also going to get water that's reabsorbed into the blood. And if we increase the amount of water in the blood, then we increase blood volume, and that increases blood pressure. So angiotensin 2 stimulates aldosterone, which stimulates sodium reabsorption, which stimulates water reabsorption, which elevates blood pressure. Another thing that angiotensin 2 does is it's a potent vasoconstrictor. It acts on the blood vessels throughout the body, and it leads to widespread vasoconstriction. And by doing that, by decreasing the space that the blood is in, we increase blood pressure. The final thing that angiotensin 2 does is it goes up to the brain, and it works on the hypothalamus. And when it binds to receptors on the hypothalamus, we get a sensation of thirst. And so we seek out water. And if we seek out water and we drink water, then that water moves from the GI tract into the blood, and that increases blood pressure. So these are 3 ways to increase blood pressure, and they're all regulated by angiotensin 2. And aldosterone is one of the mechanisms that we do that. So this just has to do with that point there. Electrolytes are involved in controlling fluid movements of the body. Sodium is the most abundant electrolyte in the body, and we find that in the form of sodium bicarbonate and sodium chloride. About 90 to 95% of all the solutes in the fluids of the body have sodium in them. And I was looking for something to illustrate, the fact that wherever sodium goes, water goes, and I actually found this little cartoon here, and this is a sodium molecule, and he's got his little pet water there. So wherever sodium goes, water follows along with it. Caffeine. Caffeine is a diuretic. A diuretic is something that makes you urinate. And the reason that it's a diuretic is because it inhibits sodium reabsorption. Okay, it prevents sodium from moving back into the blood. So if that's sodium right there, and whereas aldosterone would normally cause sodium to be reabsorbed, caffeine is coming in and saying no. All right, so what happens is that sodium stays in the distal convoluted tubule, and that means that water stays in the distal convoluted tubule. And if you have more water that stays in the nephron, then you're going to have more water that's in the urine. And this is a nice little diagram summing all that stuff up. Blood pressure falls. That's detected by the kidney. The kidney releases renin in response. What does renin do? Converts angiotensinogen to angiotensin 1. What happens next? Angiotensin 1 goes into the lungs, and angiotensin converting enzyme converts that to angiotensin 2. Angiotensin 2 acts on the adrenal gland, causes the release of aldosterone, which causes salt retention, which causes water retention, which causes blood pressure to rise. So anytime you have an axis like this that's involved in solving some problem of the body, we can manipulate it to basically to manipulate variables in the body. So if this axis right here is going to regulate blood pressure, then we can manipulate it to say try to lower blood pressure. What if you have hypertension? Hypertension is excessively high blood pressure. Well, what are some pharmacological treatments that you could do? What are some targets of this axis you could do that would cause blood pressure to go down? And the answer to that is pretty much, since this axis is involved in elevating blood pressure, anything you do to prevent this axis from doing its thing is gonna lower blood pressure. And there's a couple specific target areas here. One of those are called ACE inhibitors. And what's an ACE inhibitor going to do? It's going to prevent angiotensin converting enzyme from doing its job. And if that can't do its job, then we don't get the production of angiotensin 2. We can't convert angiotensin 1 to angiotensin 2. And if we don't have angiotensin 2, then we're not getting these effects here. We're not getting elevated blood pressure, and instead we're lowering our blood pressure. Beta blockers. Beta blockers inhibit renin. And again, if you don't have renin, then you don't have angiotensin 1. And if you don't have angiotensin 1, you don't have angiotensin 2. And we'll talk about this a little bit more, or at least remind you of it. But this mechanism we talked about is involved in raising blood pressure. And I want you to keep in mind that there's other things going on when blood pressure drops as well. We have the sympathetic response. We have the nervous system involved, which detects that low blood pressure, gets integrated in the medulla, and we increase heart rate and contractility and all that stuff in response. We act on the adrenal gland and cause the release of epinephrine and norepinephrine. We also have this low blood pressure information also goes to the medulla, to what we call the vasomotor center. And what does that do? It acts on blood vessels and causes widespread vasoconstriction. So low blood pressure is a serious problem, and we address that through the sympathetic nervous system, through these guys here, and also through the renin-angiotensin-aldosterone mechanism. All right, so all that happened in the distal convoluted tubule, and now whatever's remaining enters the collecting ducts. And at this point, that filtrate is known as urine. And the last thing that we can do to it here is reabsorb water. And like I said, that's going to be dependent on the release of anti-diuretic hormone in the pituitary gland. All right, so let's orient ourselves here. First of all, this is the pituitary gland, and that right there is the anterior pituitary gland, and you may remember that from the endocrine system. So the endocrine system, we have the hypothalamo, pituitary, and then whatever axis. And so the anterior pituitary is going to be involved in producing hormones like growth hormone, FSH, all of those hormones that are going to go down and act on downstream endocrine targets. But we have this other part of the pituitary gland called the posterior pituitary gland. And this is going to be responsible for releasing anti-diuretic hormone. Also, the hormone oxytocin is going to be released in the posterior pituitary. But this is kind of interesting, is that you have these cells up here called these neuroendocrine cells. And they're part neurons, part endocrine cells. And you have the cell body up here in the hypothalamus, and this is where, we'll use this guy. This is where anti-diuretic hormone is produced up here in the cell bodies of these neurons. Then it travels down the axons, and it's stored down here in the axon terminals in the posterior pituitary. And so when we need to release anti-diuretic hormone, it's ready to go, and it's just released from these axon terminals in the posterior pituitary. So this is the axis that has to do with anti-diuretic hormone here. And if we look at this diagram here we have the posterior pituitary that's releasing anti-diuretic hormone. But my question is, why does this happen? When do we release anti-diuretic hormone? Well, there's two main situations where we're going to increase anti-diuretic hormone. One of those is dehydration. If the body is dehydrated, we're going to release ADH. If we have a high sodium intake, too much sodium after a meal or in the body, then that's going to cause the release of anti-diuretic hormone. In either of these cases, if you're dehydrated or you have high sodium in the body, that increases the osmolarity of the body fluids. Where homeostasis is 300, maybe it raises it to 400 milliosmoles. And that's a problem, okay? That's out of balance. We need to maintain homeostasis there. We don't want that 400 milliosmoles. We have to lower it. So how are we going to do that? And you guys know, if you have 400 milliosmoles, then that causes water to move out of the cells, and the cells can shrink and cause all sorts of problems like that. So it's something that we want to solve. So when you have this increase in osmolarity here, and by the way, it says osmolality here. As far as I'm concerned, osmolarity are interchangeable. So when you have this increase in solute concentrations in the body, this increase in osmolarity, that's going to be detected by osmoreceptors in the hypothalamus. And in response to that, those osmoreceptors in the hypothalamus cause the posterior pituitary to release anti-diuretic hormone. So now we have anti-diuretic hormone in the blood. That anti-diuretic hormone is going to act on the collecting ducts of the kidneys. And what is it going to do? It's going to increase the reabsorption of water. And when we increase the amount of water in the fluids, then we're diluting out all of these solutes here. Or we're increasing the amount of water in the fluids. Either way, that decreases osmolarity and gets us back down to where we want to go. Okay, and this is how that reabsorption occurs. So here we have filtrate in the distal convoluted tubule. And when that filtrate enters the collecting duct here, its osmolarity is 100 milliosmoles. What does that mean to you guys, 100 milliosmoles? That's super dilute. Okay, there's lots of water. There's basically lots of water. And so when urine is traveling down the collecting duct, this is super dilute urine. And if anti-diuretic hormone is not released, then that super dilute urine is going to be eliminated from the body as large volumes of dilute urine. Now what happens with anti-diuretic hormone, oh, and by the way, when water is traveling down this collecting duct is not freely permeable to water. Water can't just move through here and do its thing. There actually has to be these little molecules that are inserted in the collecting duct that allow water to move from the collecting duct back into the blood. And that's exactly what anti-diuretic hormone does. So what anti-diuretic hormone does is it acts on the collecting duct and it causes the insertion of these molecules that we call aquaporins into the collecting duct. And what aquaporins do is they allow water to move from an area of higher concentration to lower concentration into the interstitial fluid and back into the blood. And by doing that, we're reabsorbing water. We're taking water that's in this dilute urine and we're moving it back into the blood. And this is just showing that process again. Here is the collecting duct, and this is a simple cuboidal cell in the wall of the collecting duct. And normally we have these aquaporins that are stored within these cells here. But when anti-diuretic hormone acts on them, it causes these aquaporins to be inserted in the walls, and again that allows water to move from the collecting duct through this wall and back into the blood. And if our osmolarity was 500 in the blood, way too high, then if we increase the amount of water in the blood, then it dilutes out those solutes and it brings it back down to where it needs to be at about 300. And now I want to put this into context with this picture of the kidney here. So to remind you guys, most of the parts of those nephrons are up here in the renal cortex. So blood filtration and tubular reabsorption is happening in these renal cortex here. But then we have the adrenal medulla. And specifically we have these medullary pyramids here. And you notice that these medullary pyramids have this striated appearance to them. Well, those striations are due to collecting ducts. Those are collecting ducts that we're actually looking at there. So what's going to happen is this urine travels down these collecting ducts And if anti-diuretic hormone is being released, then that allows water to move from these collecting ducts back into the blood. And regardless what happens is as that urine moves down these collecting ducts, those collecting ducts start to merge at this area here called the renal papilla. And what's happening at the renal papilla is that it's taking all this urine here and it's dripping it into this funnel system, specifically into the minor calyces, then they go to the major, into the renal pelvis, and then down through the ureters here. And so when you see these medullary pyramids with these striations, I want you to imagine urine traveling down there, and if anti-diuretic hormone is being released, then we're reabsorbing water from those collecting ducts back into the blood. But what about if we're not releasing anti-diuretic hormone? Well, then that dilute urine is going to continue to travel down these collecting ducts, and we're going to have large amounts of dilute urine that enter this funnel system here. And we're going to look at a couple scenarios in class that'll hopefully put this together. But one is a situation where we're dehydrated. If we're dehydrated, what's that going to do to osmolarity? What's that going to do to anti-diuretic hormone and therefore the number of aquaporins? What's that going to do to water reabsorption in the collecting duct? You know, and we'll go through this series of steps here. The other scenario is if you're overhydrated. If you're overhydrated, what's that going to do to osmolarity? What's that going to do to anti-diuretic hormone release and the number of aquaporins involved? What's that going to do to the amount and volume of urine that we're producing? So again, we'll go through these scenarios in class. Now, one last thing that I haven't talked about yet with regards to the kidney, you know, the kidney regulates water balance. It regulates electrolyte balance, which we just talked about, both of those. It's also involved in the maintenance of blood pH. And specifically, what can happen is, this is again at the collecting ducts, is the collecting ducts have the option of producing this molecule called bicarbonate. And what's going to happen is if the pH of the body is too low, it's too acidic. Okay, obviously that's a problem. And in response to that, the collecting ducts are going to produce this bicarbonate ion and ship it out into the blood. Well, how's that going to help our situation here? Well, bicarbonate binds with these free hydrogen ions. And as you guys remember, pH is a measure of the hydrogen ion concentration. So if you decrease the amount of hydrogen ions, then you raise pH back up and you make it less acidic. So again, if the body's too acidic, that means that there's too many hydrogen ions, free hydrogen ions floating around in the blood. And what the kidney's going to do in response is release bicarbonate, because what that does is that bicarbonate binds with free hydrogen and it eventually converts it to carbon dioxide and to water.